My name is Megan Keith. I'm doing my sensory physiology portfolio on the anatomy and physiology of the retina. I think it's important to start with a little definition of what is the retina. So the retina is a thin layer of tissue that lines the posterior cavity of the eye that is responsible for converting light that enters into your eye into the electrical signals that the optic nerve sends to your brain to create the images that you see. First, I want to focus on more of the gross anatomy of the retina. There are three distinct regions: the optic disc, the macula lutea, and the peripheral retina. The optic disc is a region of the retina that has no photoreceptors because it's where the optic nerve and the blood vessels exit the eye. The macula lutea is also called the fovea, is the yellow oval spot at the center of the retina that contains blood vessels and nerve fibers. This is the region that has the most acute vision and is the point in which light is focused when you look at an object. The peripheral retina is the area of the retina that surrounds the macula lutea, and it makes up the bulk of the retina. Next, I'm going to be talking about the fine anatomy of the retina and the neural cells within it. So first is the pigmented epithelium, which is the outermost layer of the retina, and this layer functions to absorb any light rays that may escape the photoreceptors, and this prevents distracting light from reflecting inside the eye and prevents them from distorting the visual image. Beneath the pigmented epithelium is a layer of photoreceptor cells, which are the neurons that convert light energy into the electrical signals. There are two different types of photoreceptor cells, rods and cones. Rods are more sensitive to light and dark, and cones are more sensitive to color. So together they work to provide you with contrast and color vision. The electrical signals are then passed from the photoreceptors to a layer of bipolar cells. Horizontal cells synapse with the photoreceptors and the bipolar cells, and the horizontal cells mediate lateral inhibition in the retina, and this enhances stimulus location and contrast. The sensory information about light then passes from the bipolar neurons to a layer of ganglion cells, but in between there, amacrine cells modulate the information flowing between them. The ganglion cells are where the signal is going to exit the retina because the axons of the ganglion cells form the optic nerve, which leaves the eye at the optic disc. Because the optic disc has no photoreceptors, images projected onto this region cannot be seen, creating what is called the blind spot. I think it's also important to touch on the neuronal circuit of the retina. Signal processing in the retina is a great example of convergence, in which multiple neurons synapse onto a single postsynaptic cell. Depending on the location in the retina, as many as 15 to 45 photoreceptors may converge onto one bipolar neuron. This increases the sensitivity to dim lights, but decreases the spatial acuity of the peripheral bipolar cell. Lastly, I'm just going to go through the steps of visual transduction in the retina. First, it starts with light passing through the lens and striking the retina, where the rods and cones become excited within the layer of photoreceptor cells and phototransduction occurs. Then, glutamate is released from the photoreceptor cells onto the bipolar cells, followed by signal conduction to the ganglion cells. This is where the optic nerve delivers the signal to the brain, and neural pathways from the retina to the brain process electrical signals into visual images that we see. For my study aid, I created a little worksheet with 3 questions, 1 matching question and 2 short answer. The first question just asks you to match the different neuronal cells of the retina with their accurate description. The second question asks about the pigmented epithelium and what would happen if we lacked it. And the last question is just about the blind spot and why it has that name.